Hello, this is video 13, DNA structure and replication. You'll notice I don't have any free response referenced. There aren't very many recent ones on structure and replication. It's good background info that you would need, so without knowing the structure of DNA, you'll end up getting into trouble on questions, but they don't come out and ask you specifically about it, typically. So I guess I would kind of compare it to, a calculus problem. They don't come out and ask you like to multiply or divide something, but along the way you have to multiply and divide to get your final answer that they ask for. So start off by looking at the structure of DNA up here. And within the structure of DNA, you'll notice that a nucleotide is always a phosphate, a sugar, and a nitrogenous base. They call it a nitrogenous base because obviously it has nitrogen, but why don't they just call it a base? Because normally when you guys think of bases, you think of something that ends in OH. Sodium hydroxide is a base. Potassium hydroxide, calcium hydroxide, those are bases. Just like a lot of times from your 10th grade chemistry, you'll think that things that start with H are acids, hydrochloric, sulfuric, phosphoric acid. But a better definition, in biology anyway, is an acid definition is something that donates or gives up hydrogen ions or H pluses. Protons easily. That's a strong acid. Things that don't give them up as easily are weak acids. If I have a base, something that's receiving hydrogen ions, that's how we would define a base. So we'll notice a few things. Whenever I first start looking at the structure of DNA, the first thing that most students notice is that A's and T's go together and G's and C's go together. And why is that though? Like why can't an A go with a G or a C with a T? Well, there's basically two rules that apply here. And that is, I need the number of hydrogen bonds to match. So A's and T's share 2 hydrogen bonds. G's and C's share 3 hydrogen bonds. That's not it though. You might say, well, why couldn't an A and an A go together or something like that? Well, or an A and a G. I know the hydrogen bonds wouldn't match up there. But also, if I have a big one and a big one, then my DNA kind of bulges out. You'll notice I always have a little guy and a big guy. Big, little, big, little, big, little, big, little. So this one, the big ones have a hexagon and a pentagon. We'll name those in just a second. The reason it matters, if my DNA bulges out or if I had like a T and a C, my DNA would kind of like concave in. That's a warning that we've got a mutation. So because DNA has a regular, repeatable, predictable pattern, proteins can slide along here, and if all of a sudden they sort of like hit a speed bump, then now that's an indication that there's a mutation and you need to stop and maybe try to fix something. Or if there's a divot inward, like that. Okay, so we don't just say big guys and little guys. It turns out the big guys are called purines. Purine. And this is so bright, I know that means that the lighting in the room is going to be dark, but what you going to do? So a purine has A and G. I think of pure as gold. And I think of gold as a big deal, so I think of the purines as being the big nitrogenous bases. And then the other guys are pyrimidines. That's, I guess I'll put the T first, since it kind of matches the A. That's T's and C's. They must be the little guys. So the more formal way to say this is A's and T's share 2 hydrogen bonds, G's and C's share 3. So the number of hydrogen bonds have to line up, and then I need a purine with a pyrimidine. Once I meet those 2 rules, A's go with T's only, G's go with C's only, and that's it. Another thing you'll notice is this sugar. This is DNA, deoxyribonucleic acid, so probably acidic. DNA is originally made in the nucleus, and it never leaves the nucleus. I guess cell division, it's leaving the nucleus. But DNA never leaves the nucleus, so nucleic. And then that sugar is called deoxyribose. In RNA, we have a phosphate, a sugar, and a base. And the difference is the sugar in ribonucleic acid is ribose. And also a difference in their bases is RNA has AUGC. Well, the difference between ribose and deoxyribose makes a whole lot of sense if you look at its structure. CH2OH, OH, OH, OH. So this up here is ribose. And if I want to make deoxyribose, I need to deoxy it. So there you go, that's deoxyribose. That's the difference between the RNA sugar and the DNA sugar. Okay, so the next thing that we notice is if I'm comparing the left-hand side and the right-hand side of DNA, one side's kind of facing up if I look at those sugars, and one side's kind of facing down. If they're both facing the same way, that's called parallel. That's perpendicular. But if they're like this, like most streets, then that's anti-parallel. So DNA is anti-parallel, sort of like you have northbound and southbound streets, right? Otherwise, I could like go to Kroger, then I could never come back. So most streets are anti-parallel. So DNA is anti-parallel. Well, calling a sugar, this deoxyribose sugar, right side up or upside down, like who's to say? Most people agree that one seems right side up because that sort of looks like a head. But if you got a more complicated molecule, how do you decide? So in chemistry, they don't refer to it as right side up or upside down. What they do is they number the carbons. So along here, this first carbon, that's carbon 1, and we'll call it, 1 prime. Carbon 2, 3, 4, and 5. So what that means is this bottom corner, like right down there, that's the 3 prime carbon. Up here, that guy, that's the 5 prime carbon. So on this side, if we're going from the bottom up, we're going in the 3 to 5 direction. 3, 5. 3, 5. 3, 5. 3, 5. Over here, if I was moving from the bottom up, I'd be going 5, 3, 5, 3, 5, 3, 5, 3. So what we would do is if I was talking with my chemistry buddies, if that's a thing, then I would basically say like, okay, I'm picturing a deoxyribose sugar and it's 5 prime facing up. And they'd be like, oh, cool. I can picture that in my brain. Or I could say 5 prime is facing down and they'd be like, okay, cool. And they can picture what I'm talking about. So that's where the 3 and the 5 come from. All right. Now, the next thing that we want to look at is the replication of DNA. And so in order to replicate, we have to unzip DNA, and we always see a picture that looks like this. And there's some truth in that, but it's also just a little bit simplified. So now what I want to do is turn the projector off and use a few drawings to kind of explain how this works. Okay, so let's say that here's my strand of DNA. And I want to replicate DNA. So on this side over here, I'm just gonna, to make it easy, I'm just gonna put all A's. In real DNA, there's gonna, be a mix and match. You can have a segment of all A's in a row, but that's not gonna be common. So I'll put all these A's right side up. And then because DNA is anti-parallel, over here, as I'm drawing the T's, I'm gonna draw the T's upside down, because they're kind of facing the other way. Again, remember, DNA is anti-parallel. All right, so whenever we want to replicate DNA, by the way, when is it that we replicate DNA? The S phase. So back in cell cycle, the previous couple of videos, we go through G1, S, G2, mitosis, cytokinesis. The S phase is when we are replicating DNA, and that's what we'll be talking about now. Well, first we have to unzip DNA, so we've got an enzyme for that. DNA helicase. And its job is to unzip the DNA double helix. So the name kind of makes sense, cool. So it's going through here and it's just sort of opening this up. You might see the term replication fork, because this is where it's kind of like a fork in the road. We split. Okay, the next thing that needs to happen, you'll notice I'm going to leave a space here. The next thing that needs to happen is DNA polymerase 3 needs to come in. So the job of DNA polymerase 3 is it's going to land on this side and start reading, and it's going to say, oh, T's, I'm going to add an A. T, I'm going to add an A. T, I'm going to add an A. Over here, when it sees A's, it's like, I'm going to add a T, I'm going to add a T, I'm going to add a T. The problem, though, is that DNA polymerase 3 can't land on single strands. So it can only land on double-stranded stuff. And so what that means is we have another enzyme here in the middle. That's why I left a space, called Primase. And its job is to lay down a little bitty piece of RNA that we call an RNA primer. Prime means to start something. You might have heard of priming the pump. I've got a gas-powered weed eater. Before I start it up, I push this little bulb thing and it squirts in like a few squirts of gasoline before I pull the cord. So we're priming. So what that means is something along the lines of, like down here, if my DNA was originally coiled like that and I'm opening up this giant replication bubble, they call it, if I put a primer down here, so let me go ahead and, those are all A's, let's say. I put down my RNA primer. Now, DNA polymerase can land on that. That's double-stranded. And then as it starts to move, it's building the second strand as it moves along. It's almost like the analogy I give is I can't take like a train engine, like a locomotive. I can't like put it on the ground and then put half a track there and have it move. But if I could have a half track all the way, this very beginning part, I could make it like a full track. I could set the train on it, and what if the train as it was driving, what if it was like building in front of itself the second half of the track? So as it drives, it builds the second half. I don't know, this might be a horrible analogy. So here we go, as my, I'm already like 20 seconds into this analogy, so we'll just see how it goes. So if I'm a train, I have to land on double-stranded track like this, and then I can move. And then I'm good to go. But if I've only got single strand, I can't land on that track. So if I could have a piece of RNA land on here, an RNA primer, now my train car could land on here. And then as I'm starting to move, I'm actually building the other half of the track as I move. So here we go. Let's do this whole analogy again. I unzip DNA so it's single-stranded. I put down a piece of RNA primer. Now the train car can land. And then as the train car is driving along, it's literally building the other half of the track as it moves. So I have some sort of mechanism jumping out in front of me, building the track as I move along it. That's what's happening. Okay, so with DNA polymerase 3, here's the third one discovered. It turns out that it reads in the 3' to 5' direction. So back over here, when we were looking at our sugars, our deoxyribose sugar, we had a carbon up here, 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 and here. This was carbon number 3. That was carbon number 3, 3', and this up here is carbon number 5, 5'. So what that means is I'm moving this way, so I'm reading 3 to 5. So think of it this way, pretend the bottom of this pen is 3 and the top is 5, 3 to 5, 3 to 5. So if I'm looking at my anti-parallel DNA, this would be parallel DNA, but DNA is anti-parallel, so it's like this. So this side I'm going bottom, top, bottom, top, bottom, top, 3, 5, 3, 5, 3, 5. So that means if I'm reading in the 3, 5, 3, 5, 3, 5 direction, I need to be writing bases, I need to be putting down on the other side 5, 3, 5, 3, 5, 3, because DNA is anti-parallel. So I read 3 to 5, and I write 5 to 3. That's why if you just say something like, DNA polymerase 3 works in the 3 to 5 direction, you're like, what do you mean it works? If I read 3 to 5, then I write 5 to 3. So now I'm cruising this way, and I'm reading 3 to 5 on the purple strand, which means I'm putting down in the 5 to 3 on this strand. So here's my piece of RNA. I'll cruise along here, my primer is down, my DNA polymerase 3 lands, and here I go. So I'm just matching these things up. If there's A's, I'm putting down T's. All right, great. The problem is that because DNA polymerase reads 3 to 5, that means that I've got one DNA polymerase 3 going this way, and I'm going to have another one. Let's say I put down a piece of primer here. I'll have another one that landed and is going this way, right? Because if I look at my purple strand, 3 to 5, that means this way on the purple strand is 3 to 5. So I have to be going this way. So it's cruising along and it's adding bases going this way. You're like, all right, that's not a problem, Mr. Cotton. I've got one polymerase moving here and one moving here. But there is a problem because they're connected. So I have to be able to have one going up and one going down at the same time, but they're connected. So how is that possible? Well, I made these. So here's one side of DNA. It says 5' on there. I'll put a 5' on this one too. Either way, the blue side is the 5 side. The side without blue is the 3 side. So this would be parallel. That's not the case. It's anti-parallel. And ignore that. That's where the string got damaged. I was gonna start a GoFundMe, but so anyway, I'm like this. So here's my purple side, 5 at the top. Here's my purple side over here, 3 at the top. So the way that it works is one side is what they call the leading side. It's going this way. I put down a piece of primer, I take off. If I unzip more DNA up there, I just keep on going. So here's my DNA polymerase 3. It lands here and takes off. Right now it's stuck because I haven't unzipped more. When helicase unzips more, I just keep on going. However, on this side, it's different. On this side, what happens is I landed right here, my DNA polymerase 3 lands here, and it starts moving downwards. Once I unzip more, now my DNA polymerase is down here. So once I unzip more, let's say I unzip a little bit more DNA up here. Then now, to copy that side, I've got to put down another piece of primer. So Primase will come in and put down another piece of primer. And then my DNA polymerase 3 has to unlatch and grab on there and then build this strand. So this is the leading strand. This over here is the lagging strand. And on the lagging strand, you'll have like a piece of primer, some DNA, a piece of primer, some DNA, a piece of primer, some DNA, all along here. Here's a piece of primer from earlier when we were just unzipping this DNA. And so we have these like islands, these fragments of DNA all along the way on the lagging strand, and those are called Okazaki fragments because Okazaki discovered them. But again, we still have this problem of here's my DNA, and I've got 5' here, I've got 3' here, and so somehow I have to have one of my DNA polymerases going down as the other is moving up, but my hands have to be connected. So how do I do that? Well, pretend that DNA is double helix together and pretend that my head is helicase. So I'm unzipping this. Okay, so here we go. This is 3. So DNA polymerase lands on some primer and just starts cruising this way. Great. Now there's a primer right here. This is my 5. So towards the blue is the 5. So I have to move towards the blue. This guy is moving towards the blue. This guy is moving down towards the blue. So my head unzips, DNA helicase, and now I start going towards the 5. This one starts going towards the 5, but my hands have to be connected. So how do I do that? Well, whenever I unzip more, now what I have to do is I actually flip. So if I unzip more DNA, if I flip this around, now this guy can go toward the 5. Now they're both going, they're reading from 3 towards the 5. So this one's going towards the 5 and this one's going towards the 5. I unzip more DNA, I flip it around, 3 to 5. Unzip more DNA, flip it around, 3 to 5. But you'll notice this hand, as I unzip more DNA, I latch down on one primer and then I just never have to let go. So this side over here will have one primer at the very, very bottom, and that's it, all the way up. This one, because I'm having to put down a primer, read some, flip it, put down a primer, re-grab, read some, flip it, put down a primer, re-grab, read some. So this, here I have like primer, DNA, primer, DNA, primer, DNA, because I'm having to flip this other side, the lagging side. And so that's why you're seeing this over here, is because really the DNA polymerases are like joined together and we're flipping the DNA as we go. I shut down my projector, but I do have a picture that I'll show you here really quick. I'll turn the projector back on. Super professional video. But hey, how many teachers do you know that have projectors at home? So in this video, they're going to actually kind of show you what this looks like. In fact, it might even be easier to just have you look at my computer screen. Since we're waiting, if you look at my screen, you can actually see that on the leading side, everything is pretty easy. On the lagging side, I have to constantly like flip that strand around. Okay, now, the next thing is that, so I think I will actually turn that off. So the next thing that we wanted to emphasize is, okay, I can't leave these pieces of RNA in here forever. So we've got another enzyme, DNA polymerase 1. His job is he sort of lands, he also reads in the 3 to 5 direction, but he lands, I'm just going to say for easy terminology, ahead of the primer. So he'll land ahead of the primer on the 3N side, kick out the primer, put in new bases. He'll land ahead of this primer, kick out the primer, and then add in, he backs up, and then adds in new bases. That's great. Except over here on this end, there's a problem on that end. I can't land above that. Same thing here. I'll have a primer down here on this 3' end. We'll focus on this one, but I'd have the same problem on the opposite strand over here. So now my DNA polymerase has to land above that red part and then move down. But you can't land above the red part because there's nothing there. And so because of that, every time our DNA replicates, we aren't able to kick out that primer. When DNA and RNA stay locked together too long, they get degraded. So after a while, that gets degraded. Same thing on this end. So every time my DNA is replicated, I lose a little bit of DNA off of the ends. And so what did we have as kind of a way of, avoiding catastrophe? Telomeres. If I've got DNA and every single time my DNA divides, I lose some bases off of the end, well, if those bases coded for like normal heart or lung or liver production or something like that, then I'm screwed. So what my body does, or what my cells do, is it just puts in telomeres, just segments of garbage. So telomere is just the segment TTAGGG. It's just filler. So on the ends of my DNA, I just have tag, 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 tag. So whenever my DNA replicates and I lose some bases off of the end, it's just filler, it's tag. The example that I gave you guys in class was, let's say that I wanted to copy this book, like on a copy machine. But every time I put it on the copy machine to make copies, I lost the first page and I lost the last page. Okay, well when I lose this blank page here and this blank page here, it doesn't really matter too much. But pretty soon when the cell divides again, every time I copy my DNA, I'm losing pages. Before long, I'm like missing out on actual important stuff. So what could I do? I could just put a few thousand pages of crap in the front and a few thousand pages of crap in the back. And now it doesn't matter. Blank pages, gibberish pages, I don't know, social studies. And so then every time I divide, if I lose something, nobody gives a crap. And that's what telomeres are for. Because I'm unable to replicate the very, very ends of my DNA. So DNA polymerase, again, whenever it will go in and like add in new bases, it just joins hydrogen bonds. So this DNA polymerase 1 that will remove primers and add in DNA, that's why it's a DNA polymerase. It's pronounced polymerase. I always think of as a teacher, I'm always saying polymerase because it's a polymer enzyme. It's an enzyme that makes polymers of DNA. So when DNA polymerase 1 goes in and puts in the new bases, it doesn't seal the edges. So we have one last enzyme. Ligature. What's a ligature? It's like tying together. So ligase is an enzyme that will seal the edges. So it's joining the phosphate sugar bonds along the way, the phosphodiester bonds along the way. DNA polymerase is just joining the A's to T's and G's and C's, and then ligase seals up the edges. Once we're done with that, our DNA is now replicated. What's weird about it though, and again, I'll have to kind of just sort of ghetto show you on my computer, is all of these enzymes are globbed together in one big complex. So it actually looks, you guys might remember this video, like this. So here's my DNA going in, here's the leading strand, here's that lagging strand on top. The lagging strand is the one on top, there we go. The lagging strand is the one on top that's having to flip around. So they're going to emphasize, there's the lagging strand. Have to kind of flip it around and copy it backwards. So if I kind of fast forward into this a little bit, I can see helicase doing the unzipping. And then on the back end, I'm going to see those DNA polymerases adding in the nucleotides. All right. Our next video will be transcription, and then we'll get into translation and then gene regulation. So thanks for coming out.